Assalamu alaikum dear students, welcome to your channel The Anatomy Canvas, and in today's class I am going to discuss with you the development of the musculoskeletal system. So learning outcomes for the today's class are classification of the mesoderm, the intramembranous and endochondral ossification, limb development, ossification of bones of the limbs, development of joint and cartilages, developmental events of fibrous joints, of cartilaginous joints and of synovial joints, then the important congenital anomalies and development of the muscular system. So all that stuff which is related to development of the muscular system we will cover in today's class. First of all, we will do the classification of the mesoderm. So as you know from your previous knowledge that the mesoderm is the tissue which migrates from the area of the epiblast and comes to lie between the ectoderm and endoderm and form the mesoderm. This mesoderm then differentiate into four different types of the tissue. The carda mesoderm, which will form the notocard, the paraxial mesoderm, the intermediate mesoderm and the lateral plate mesoderm. This lateral plate mesoderm again differentiate into two, the somatopleuroc layer and the splanchnopleuroc layer. Here in this diagram also you can see this is the neural tube. This area is the cardomesoderm forming the notocard. This is paraxial mesoderm, the intermediate mesoderm and the lateral plate mesoderm forming the splanchnic layer and the parietal layer. Now the derivatives, the carda mesoderm will form the notocard, the paraxial mesoderm will differentiate in the head region into somitomeres and the occipital region caudally into the somites. These somites will form the sclerotome, the myotome and the dermatome. The sclerotome in turn will form the cartilage, the bone, the muscles, cartilage, bone, the tendons, the ligaments of the area, and the myotome will form the skeletal muscle and dermatomes will form the dermis. Then the lateral plate mesoderm will form the splanchnic mesoderm, somatic mesoderm and also contribute to the extra embryonic mesoderm. Similarly, the intermediate mesoderm, which is present between the paraxial mesoderm and lateral plate mesoderm, this is intermediate. It will develop into the kidneys and the gonads. So these are the different mesodermal derivatives which will form different tissues in human body. Now the sources of the skeletal system development, very important MCQs regarding sources of skeletal system development. Most important, the paraxial mesoderm, also the part of the lateral plate mesoderm and to some extent the neural crest cells. So these are the three main sources from this which the skeletal system basically develops. So now we move on to the process of the somitogenesis. So somitogenesis process, it takes place that the paraxial mesoderm first form a compact mass. The process is called compaction. After a compact mass is formed, then there are the changes from this mesenchymal to epithelial transition. Epithelialization occur and a donut shaped structure is formed. After this donut shaped structure is formed, this is called the somitomeres in the head region and somite from the occipital region caudally. After the formation of the epithelialization, again changes occur in which the cells which are present over here in this border, they will again show from epithelial to mesenchymal transition. They form loosely woven tissue and these cells migrate from this area and surrounds the area of the notocard and neural tube and they will form the sclerotome. The cells which remain over here which form the dermomyotome. So after the sclerotome formation, the dermomyotome, the dorsomedial edge will form the cells which are called the epaxial myotome. It starts from the dorsomedial edge and also the ventrolateral edge cell present at the ventrolateral edge. They also become myoblast and they form the hypaxial. The epaxial also called primaxial and hypaxial also called the abaxial. The remaining cells which are shown in green color over here, they form the dermatome. So in this way, whole of the somite will differentiate into the sclerotome, the myotome and the dermatome. So this is the process of the somitogenesis. Again in this diagram, this is a diagram from your Langman's embryology. Here you can see this is the area of the notocard. Then you can see the area of the neural tube. Then the notocard present over here. Here you can see the termomyotome, the sclerotome, the two edges, dorsomedial edge and ventrolateral edge. Again must remember some of the cells from the ventrolateral edge, they migrate and they crosses a border. This border is called the lateral somatic frontier. These cells from ventrolateral edge migrate, they cross the lateral somatic frontier and they form part of the somatic layer of the lateral plate mesoderm. And this is very important. This lateral somatic frontier is very important because for the signals for the development are from this area are derived from the lateral plate mesoderm, while signals for development of the dorsomedial group they are derived from the neural tube and the notocard. Second, the dermatomal pattern is also different. The ventrolateral the cells which migrate from the ventrolateral edge and they crosses the lateral somatic frontier, they will form the dermis in the body wall anteriorly and the limb, and the cells which are present over here in this area, they will form dermis of the back. Similarly, area for the rib development is also different. So the cells which are formed from this dorsomedial edge, they will form the bony component of the rib, while the cells which are migrating from this here, they will form the cartilaginous component of the rib, which will attach the rib to the sternum. So this lateral somatic frontier is very important and it defines the borders. Then the different factors which are involved in the somitogenesis. As I have told you, the cells which are derived from the ventrolateral edge, they crossing the lateral somatic border, their pattern is determined by the factors secreted from the lateral plate mesoderm. So this factor is the bone morphogenetic protein 4, which affect the myoblastic cells and affect and stimulates the myogenic factor D. Also the WNT is released from the surface ectoderm, which also stimulates the myogenic factor D, and as a result of activation of the myogenic factor D, the cells which are present over here, they will form the myoblast. Similarly, the cells which are present at the dorsomedial lip of the termomyotome, they will stimulated by the sonic hedgehog protein, and which is secreted by the floor plate of the neural tube and the notocard. Also the WNT protein, which is secreted by the distal part of the neural tube, and this is activated by BMP4 again from the surface ectoderm. So this WNT and sonic hedgehog, these two proteins activate the myogenic factor F5 in this dorsomedial lip, and as a result of its activation, this area will differentiate into myoblast. The remaining cells in this area, they are again induced by neurotropic factor 3 from the neural tube, which will stimulate the PAX3, and this will form the dermis. So these are the different neurogenic factors which are involved in the formation of the process of the somitogenesis. Next learning outcome was about the intramembranous and endochondral ossification. So first we will see the intramembranous ossification. So have a look at this diagram. These are smaller cells. These cells are the mesenchymal cells which are present, and surrounding fibers are the collagen fiber. So the area where intramembranous ossification will take place, first of all the mesenchymal cells over here, then they differentiate directly into osteoblast. So this is very important point, because in intramembranous ossification, mesenchymal cells directly differentiate into osteoblast. First they become the osteoblast. After their formation into osteoblast, the osteoblast starts secreting the osteoid, and this osteoid will be mineralized within a few days. So after the mineralization of the osteoid, the osteoblasts which are present in the center, they are entrapped and they form the osteocytes. The third step is that the blood vessels they invade the central area, and the bone which is forming will form irregular bony plate or trabeculated bone which is present. The mesenchymal cells which are present at the periphery, then they condenses and they form the periosteum. After this, In the fourth step, bony color of the compact bone forms and the red marrow appear. So in the area which is the outer area, here the compact bone will be formed, and in the center the trabeculated bone will be formed, and cavities will be present in which blood cells and blood vessels will present, and this will form the bone marrow. So these are the four different steps in which intramembranous ossification takes place. First, again I repeat, first is ossification center appear in fibrous connective tissue with formation of the osteoblast. In second step, osteoid which is secreted is then mineralized, after which woven bone and periosteum form, and later on bony color of the compact bone forms, and in the center red marrow cavities appear along with the trabeculated bone in the center. Next is the endochondral ossification. So endochondral is different from the intramembranous in a sense that in case of endochondral, first an cartilage model will be formed. So here you can see a cartilage template or model is formed. After formation of the cartilage model, cells in the center of the cartilage, they hypertrophy and they form the chondroblast. After the formation of the chondroblast, next step will be take place, and these steps will be in which the primary ossification center appear. The bony collar will start to appear, and this will be the first step in formation of the ossification. This is the first step in ossification by the process of the endochondral ossification. So when bony collar forms, also the blood vessels invade this area, and by invasion of this area, the cartilage matrix which was formed by the chondroblast now will be replaced by the osteoid, and osteoid will be formed by the osteoblast, and these osteoblasts will come along with the blood vessel into the area of the developing bone. So as the cartilage matrix is replaced by the osteoid, the chondroblast will start to die, and they will disappear, and the instead of the chondroblast, the osteoblast which takes place, and the bone lengthening starts. So these are the different stages which are present in prenatal life, and in the postnatal life the secondary ossification centers also appear in the epiphyseal area. Here you can see the four phases which are present in the process of the endochondral ossification. First zone is the zone of the cell division, in which the cells are added both for the interstitial as well as the oppositional growth. After the cell division, the zone of the hypertrophy is present. Then the area of the calcification, and then area where the after the calcification, the chondrolysis will take place. Here in this third area, the chondrolysis will be present. The vascularization and osteogenesis will take place, and after this, in the fourth area, bone remodeling will take place, in which osteoclasts which are coming from the along with the blood vessels in this area, they will remodel the bone, and this will be the last step which usually occurs at adult stage, in which the length of the bone has completed its development in lengthwise, and their epiphyseal cartilage will completely ossify, and it will be shown by the epiphyseal line, and this occurs usually at the adult stage under the effect of different hormones, after which no increase in bone growth is possible. Again, in this diagram you can see the different stages. So first stage is formation of the bone collar around the hyaline cartilage model. Second is cavitation of the hyaline cartilage along in which the cells hypertrophy and then they deteriorate, and in third step blood vessels appear, and along with the blood vessels the osteoblast appear and osteoid will be laid down. So these three steps usually take place in fetal life from the week 9 through 9 months. This is very important MCQ. Then stage 4 usually take place after birth. Just before birth, stage 4 usually take place just before birth, in which the secondary ossification center appear, and stage 5 is the process of the long bone growth during childhood and adolescence, and at the age of the adulthood this bone will ossify, after which no more increase in bone growth will be possible. So the different steps, initially hyaline cartilage model will be formed, then the first sign will be cuff of the bone around the cartilage model. With the establishment of the periosteal bony color, the chondrocyte in the mid region will become hypertrophied. Later on, calcification taking place. Along with the calcification, death of the chondrocytes take place and osteoblast starts to secreting osteoids, and after that osteoclast causes the bone remodeling. So these are the different steps in which by the process of endochondral ossification, in which initially a cartilage model will be formed which will transform into bone. So, model in hyaline cartilage is called cartilage model, gradually replaced by bone, begins late in the second month of development. Pericardium is invaded by vessels and becomes the periosteum. Osteoblast in periosteum lay down collar of the bone around the diaphysis. Calcification in the center of the diaphysis occur, and primary ossification centers are present in all long bones at 12 weeks. This is a very important MCQ, that the primary ossification center they are present in all long bone at 12 weeks. Again, you must remember that each long bone has the multiple secondary ossification center as well. Usually 2 secondary ossification for the long bone, usually one secondary ossification center for the short bone, and many 1 to 2 or more than 2 ossification center, secondary ossification center for the irregular bone. So primary is present at 12 bone, secondary is present in epiphysis usually after birth, except for distal end of the femur and proximal tibia, which appear during 9 months. This is again another MCQ. The epiphyseal growth plate closes at the end of the adolescence. Diaphysis and epiphysis fuses and no more bone lengthening occurs. This one is the histological diagram, in which you can see this is the area where the cells undergo mitosis. The older cells enlarges, the cells are hypertrophy, then calcified, then cells die, cartilage cells die, and after which the ossification and bone remodeling take place. This is again another diagram in which you can see this is the area in which the bone formation is taking place, and here you can see the bone lining cells which are present just in near to the osteoid formation. These are the osteoblasts. This is the slide of the endochondral ossification, in which you can see area of the reserve hyaline cartilage, area of the cell division, area of the hypertrophy which is present, then the area of the calcification of the osteoid, area where the chondrocytes, death of the chondrocytes take place, and then modification and remodeling of the bone formation occurs. Next learning outcome is the limb development, and in this topic we will also cover the development of the joints, cartilages, and all types of the events in fibrous, cartilaginous and the synovial joints. So first of all, the important MCQ over here is the cells of the origin of the limb part. The most important are the paraxial mesenchyme of the somoid, the myogenic cells of the muscles which are present over here, and also the somatopleuric mesenchyme which is forming connective tissue of the limb, including cartilage, bone, tendon and loose connective tissue. The two main important sources are these. Then the angiogenic mesenchyme, which is forming an extensive vascular network in early limb bud, and along with this motor and sensory nerves which will innervate this area, and also the neural crest cell which will form the Schwann cells and melanocytes of the skin. So here in this diagram you can see this is the ectoderm. In the center you can see the mesoderm, and here in yellow color you can see the endoderm. Here you can see the formation of the neural tube. Here you can see the notochord which is forming. This is the paraxial mesoderm, the intermediate mesoderm and lateral plate mesoderm. In this area you can see the upper limb bud and the lower limb buds which are forming. So limb development begins with activation of the group of the mesenchymal cells from the somatic layer of the lateral mesoderm. Limb buds first appear as elevation of the ventrolateral body wall towards end of the fourth week opposite the somite 8 to 10. So here you can see this is the usually at the end of the fourth week, and upper limb buds usually appear at day 26 or 27, and lower limb buds appear a little later, either 1 or 2 days later. So these are again your MCQs. Then the limb bud elongate by proliferation of the mesenchyme. The upper limb bud appear low on embryo's trunk because of early development of the cranial half of the embryo, and upper limb bud develops opposite the lower 5 cervical and upper 2 thoracic, that is the origin for the brachial plexus, and lower limb buds forms the opposite the lower 4 lumbar and upper 2 sacral, that is the origin of the lumbosacral plexus. So at day 32, upper limb buds are pedal shape and lower limb buds are the flipper shape. Then a very important concept you must understand during development. First we will study there are the 3 axes which are forming. So in these 3 axes, the first of all, the main important axis is the proximo-distal axis. So elliptical, this one is the elliptical base of the limb bud, and the axes are forming from the center of this this elliptical base through the area of the mesenchymal core to the center of the AER. So this AER is actually area of apical ectodermal ridge. These are highly columnar cells which are present over here. These are constantly dividing cells which constantly give cells to this area, and the line passing from the center of this area passing through mesenchyme to the center of AER determines the proximo-distal axis of the limb bud development. Next are the craniocaudal axis. So the line, the border which is cranial to this developing area, and it is also at 90 degree to the proximo-distal axis. So this line is called the pre-axial border, and this one is called the post-axial border. And axes which are forming from pre-axial to post-axial border are called the craniocaudal axis. Then the third axis, which is from the dorsal to the ventral, are called the dorsoventral axis. So these are the 3 different axes which determines the area of the limb development. The AER basically defines the proximo-distal axis. Another important thing which you can see over here is the zone of the polarizing activity. These are the cells which are present at the post-axial border, the very important cells which maintains the anteroposterior axis, craniocaudal axis of the limb development, and also maintains the number of the digits and development of the thumb. Then the dorsoventral axis, also called the anteroposterior axis, is formed, and they will develop the surface ectodermal covering will define their development. So these 3 axes are very important. Zone of polarizing activity is a small region of the somatopleuric mesenchyme on the post-axial border. Here you can see in this area, this is the small region of somatopleuric mesenchyme on post-axial border. It has been suggested that digit number is controlled by regulation of production of apical ectodermal ridge maintenance factor, while digit pattern is regulated by the production of diffusible morphogenes that act in concentration gradient pattern. So these are the different genetic factors which are responsible for number maintenance for the digit pattern, and also for the production of the apical ectodermal ridge. So apex of the each limb bud, here again you can see this apex, this here the ectoderm thick, it become highly columnar, it form the apical ectodermal ridge. This ridge exerts inductive influence on limb mesenchyme to initiate growth and development of the limbs in proximo-distal. So this is very important. The cells which are present near to this apical ectodermal ridge, they will constantly divide, while the cells which are farthest from this area, or in other words, which is in the proximal part of the limb bud, they will start differentiate into muscles, bones, cartilage, and all the other elements. So initially a constriction is present between the hand plate and the proximal part. Later on another constriction is present, and this will divide the two area into the proximal area, the arm area, or in the lower limb it will form the femur, and in upper limb it will form the humerus. This area will form into radius ulna in upper limb, and in lower limb will form the fibula and the tibia, and this area will form in upper limb the carpal and metacarpal, and in case of lower limb it will form the dorsals or metatarsal. Again, another MCQ is zone of polarizing activity which is present at the post-axial border derived from somatopleuric mesenchyme. It controls patterning of AP axis of the limb, digits number, digit pattern through the secreted factor which is sonic hedgehog. So this is again another important MCQ over here. Then the distal end of the flipper-like limb buds flattens into paddle-like hand and foot plates, and by end of the 6th week, the mesenchymal tissue in the hand plate has condensed to form digital rays. Interval between digital rays are occupied by loose connective tissue, and these mesenchyme breaks down, forming notches between the digital digits. So here you can see initially these foot plates and hand plates, they are flattened. Later on they are webbed shape, and after this the programmed cell death occur in this area, which will later on form the fingers and the toes in upper and lower limb respectively. Again, the same diagram. Here you can see the apical, ectodermal ridge and development of all these areas. Again, almost the same diagram, upper limb development and lower limb development, the paddle shape area. Here you can see the flipper shape area, the webbed hand and feet, and after these separate digits are present. So at the tip of each digital ray, a part of apical ectodermal ridge induces development of the mesenchyme into mesenchymal primordia of the bones, that is phalanges and the digits. So blocking of the cellular and molecular apoptosis could account for syndactyly webbing or fusion of the fingers. As I have told you that these areas are webbed shape initially, and if this programmed cell death do not occur, there may be syndactyly or fusion of the fingers and toes. So in most bone, like the long bones in the limb, condensed mesenchyme which develops from the mesodermal and neural crest cells undergoes chondrification to form initially hyaline cartilage model. The chondrification centers appear later in 5th week, and by 6th week the entire limb skeleton is cartilaginous. So important point here, you must differentiate between the two things. The cartilage model is present at end of 6th week, but primary ossification center will be present at 12th week. So these are very important MCQs. From the ventrolateral region of the dermatome of the somites, the myogenic precursor cells migrate into limb bud and later differentiate into the myoblasts. As I have told you, as long bones form myoblasts aggregates and form large muscle masses. So in general, this muscle mass separates the dorsal and ventral aspects are the components. The mesenchyme in the limb buds give rise to bone, the ligaments, the blood vessels. So most of the bones of the appendicular skeleton is derived from somatopleuric mesenchyme, but body and spine of scapula is derived from somite. This is again your MCQ as well, that in case of the appendicular skeleton, somatopleuric mesenchyme will form all of the bone except the spine of the scapula and its body, which will form from the somite. The cervical and lumbosacral myotomes contribute to muscles of the pelvic and the pectoral girdles. Now another important point, another MCQ, after the formation of the initial limb bud, in the 7th week of the development, the limb extends ventrally. The developing upper and lower limb rotates in opposite direction and to different degrees. So upper limb buds rotate 90 degree laterally in such a way that the limb Thumb is positions at the lateral aspect, and the anterior compartment is flexor compartment and posterior compartment is extensor compartment. While in case of the lower limb, it rotates 90 degrees medially, as a result of which anterior compartment in lower limb will be the flexor and the posterior compartment in lower limb will be extensor, and big toe will be present on medial side. So this is the rotation of the limbs both in upper and lower limb, and their direction is different to each other. Embryo about 52 days, the fingers are separated and toes are beginning to separate. Note that feet are the fan shape. So here you can see the fan shape feet. And 56 days embryos, all regions of limb are apparent, and digits in hands and feet are separated. Radius and ulna, thumb and crude toe, they are homologous to each other, they will be formed. Next is very important, the development of the joint. So area where the joint is going to be formed, here in this area the chondrocytes which are formed, they will form the interzonal mesenchyme. And after the formation of the interzonal mesenchyme, this mesenchyme will initially differentiate into dense fibrous connective tissue. This point is very important. So initially the interzone is formed, and after this interzone, this mesenchyme will differentiate into dense fibrous connective tissue. Then the fate of this dense fibrous connective tissue is different in all the three joints. In case of the fibrous joint, this dense fibrous connective tissue remains there and it will develop into fibrous joint. In case of the cartilaginous joint, mesenchyme between these two areas differentiate into cartilage, which will form the articular cartilage in these areas. And in case of the synovial joint, a synovial cavity will be formed as within the intervening mesenchyme by breakdown of the cell. So in the center you can see breakdown of the cells which take place, and as a result a cavity will be formed. So mesenchyme give rise to synovial membrane and capsular and other ligaments of the joint. So for undergraduate levels, you must know initially the interzone will be formed after this dense fibrous connective tissue. So this dense fibrous connective tissue in case of fibrous joint will remain fibrous in nature. In case of cartilaginous joint, it will become the cartilage, and in case of the synovial joint, the cartilaginous cells in the center, they will break down and will create a cavity, and mesenchyme will differentiate into the capsules and all other elements of the synovial joint. So again another diagram in which you can see here the apical ectodermal ridge, initial the first constriction and then the second constriction where the joint area will be formed. The important point over here is the blood supply of the limb. So you must know that from the dorsal aorta, the dorsal intersegmental arteries arises, which supplies both the upper limb and the lower limbs. So this dorsal intersegmental artery breaks down into a fine capillary network into mesenchyme, which will then drain by its tributaries and which will later on form the veins. So in case of the upper limb, the main artery, which is also called the axis artery, this axis artery in arm is the brachial artery, and in case of the forearm, this is the common interosseous artery, and they will break down into the capillary network, which will then form the tributaries, the venous system, and will drain into cephalic and basalic vein. In case of the lower limb, this dorsal intersegmental artery in upper part, it will be shown as the inferior gluteal artery. This is the axial artery of the area of the thigh, and in case of the leg, the axial arteries are the anterior and posterior tibial arteries. So this is about the blood supply of the limb. Then the last outcome is the second last outcome is the congenital correlates. So congenital correlate, very important element is the thalidomide. This is absolutely contraindicated in women of childbearing age because it is main factor, and its exposure before 33 days may cause a large number of the limb developmental anomalies. So here in this condition you can see it is called the quadruple amelia. In this condition both the upper limbs and the lower limbs do not develop. This is a condition also called the meromelia, in which the absence of upper limb bud is present. It is just represented by the rudimentary stump. This is again meromelia, in which this rudimentary stump is directly attached to the trunk. So important MCQ about this congenital congenital anomaly is the thalidomide. Then another is critical period of the limb development is from 24 to 36 days after fertilization. This is another very important MCQ. Then next, the different anomalies which will be present. Major anomalies appear in about 2 in 1000 newborns. Most have genetic factors. Molecular factors have been implicated as well. Several congenital anomalies of arterial system may be present. So here in this case you can see the syndactyly, in which the fusion of the digits is present. The polydactyly, in which the extra amount of the extra number of the digit is present, or ectrodactyly, in which absence of the digits occur. So these are the different anomalies related to digits. Another very important and very common is the congenital club foot. Any deformity of the foot involving the talus is called the club foot or talipes. There are different varieties of the talipes. It is an uncommon, occurring about once in 1000 births. As the children develops, they tend to walk on the ankle rather than their sole of the foot. So out of these club foot, talipes equinovarus is the most common type, and it occurs twice in males. In this condition, the sole is turned medially. Here you can see it is inversion of the foot with hyperextension and incurving of the feet. So this is talipes equinovarus. It results from abnormal position or restricted movements of the fetus lower limb in utero. The hereditary factors and environmental factors are also important. Clubfoot appears to follow multifactorial pattern of inheritance. The interaction of the genetic as well as environmental factors both occur. Next is the osteogenesis imperfecta and then the Holt-Oram syndrome. So in case of the osteogenesis imperfecta, there is shortening and bowing and hypomineralization of the long bone with blue sclera. Genes are involved in production of type 1 collagen. So these all are your MCQ. Please remember, in case of osteogenesis imperfecta, if MCQ is present, there will be also clue about the blue sclera along with bowing and hypomineralization. And third, there will be type 1 collagen, which is important. It will be affected. In case of the Holt-Oram syndrome, the upper limb deformities with the heart defect and hypospadias in males and the biconjugate or didelphic uterus in females occur. So here in this you can see the Holt-Oram syndrome. Here you can see the upper limb anomaly in case the radius is affected. Then the Marfan syndrome, the mutation in fibrillin gene which is present on 15q, the 21 focused at one location. So the tall and slender long bones will be present, long thin face, the pectus excavatum or pectus carinatum. So this is the condition in which the fibrillin gene is important. This is Marfan syndrome. Important MCQ related to Marfan syndrome is location and the gene which is important, that is fibrillin. Craniosynostosis radial aplasia syndrome, in which you can see there is premature closure of the sutures in the cranial cavity along with aplasia of the radius. The last outcome for today's class is the development of the muscular system. So muscular system consists of the skeletal muscle, the cardiac muscle, the smooth muscle. The skeletal muscles are derived from paraxial mesoderm, and this will form the somitomeres in the head region and somites from occipital region to the sacral areas. Smooth muscle cells differentiate from the splanchnic mesoderm surrounding the gut and its derivative. The exception to this rule is the pupillary muscle, the muscles of the mammary gland, and the erector pylori muscle of the sweat glands, which are derived from the ectoderm. So these are very important MCQs. Thus, all of the smooth muscles, they differentiate from the splanchnic mesoderm, except these three smooth muscles, which develop from the ectoderm. The cardiac muscles, they differentiate from the splanchnic mesoderm surrounding the heart tube. So first, again, another slide for a revision purpose, that this is the somite, which is differentiating into the sclerotome, the myotome areas, and the dermatome. So this is the dorsomedial edge, the ventrolateral edge, the lateral somatic frontier. This is a somatic layer of the lateral plate mesoderm. So another important point here, you should must know that because of this lateral somatic frontier, the two domains are formed, either the premaxial domain or the abaxial domain. So cells which will remain in this area, which will not cross the lateral somatic frontier, they will form the premaxial domain. And the cells which migrate from ventrolateral edge, they cross the lateral somatic frontier, they will form the abaxial domain. This is a very important and a new concept of the development of the muscles. Previously, it was thought that muscles were categorized into two classes, either as the epimere or the hypomere, or epaxial or hypaxial, that is below the axis and above the axis. And because of the reason that one of these were supplied by the ventral primary rami and other were supplied by the dorsal primary rami. But because of the embryologic origin, these are now classified as the premaxial, which are derived from the dorsomedial edge, and the abaxial, which are derived from ventrolateral crosses lateral somatic frontier and combined with the lateral somatic frontier. So these are the abaxial. So cell derived from VLL will form the infrahyoid muscle, the rectus abdominis, internal and external oblique, and limb muscle. And remaining muscle, which are the premaxial, they will form the muscles of the back, shoulder, and the pelvic girdle, and the intercostal muscles. So again, almost the same diagram. Patterning of the muscle, again it is very important. Pattern of the muscle formation is controlled by connective tissue in which myoblasts are developing. So in the head region, this connective tissue is derived from neural crest cells, which will determine the pattern of muscle formation. In cervical and occipital region, the connective tissue is derived from the somatic mesoderm. And in body wall and limbs, again this connective tissue is derived from the somatic mesoderm. So head musculature, basically the skeletal muscles in the head area, they are derived from the paraxial mesoderm, including muscles of the tongue and eye and the muscles of the pharyngeal arches. They all are derived from paraxial mesoderm. Only exception is the muscle of the iris, that is derived from the optic cup. This is again your MCQ. So this is about origin of the muscles from abaxial and premaxial. The abaxial, which have migrated, and premaxial, which are present at dorsomedial edge. So in the case of the cervical region, premaxial will form the scalenus muscle, the geniohyoid, and prevertebral. And in case of abaxial will form the infrahyoid muscles. Then the thoracoabdominal region, only intercostal muscle will be formed from the premaxial. All other muscles, that is pectoralis major, minor, external, internal, oblique, transversus abdominis, sternalis, rectus abdominis, and pelvic diaphragm as well. These all are derived from the abaxial muscle precursor, which have migrated from the ventrolateral edge. Then in case of upper limb, the rhomboid, levator scapulae, and the latissimus dorsi. Only these three muscles are derived from the premaxial. All other upper limb muscles are derived from distal limb muscles, and in case of the lower limb, all the muscles of the lower limb are derived from the abaxial. Innervation, again very important. New description of the muscle development, whether the premaxial and abaxial, is different from the old concept, the epimere and the hypomere, which was based on the innervation. New description is based on actual embryological origin of the muscle. Apexial mean above the axis muscles that are innervated by the dorsal primary rami. So here in this case, you can say this is the dorsal primary rami, and this is supplying the external muscle. These muscles are called the apexial. The muscles which are supplied by the ventral primary rami, they are called hypaxial or below the axis. This concept is still present because the nerve supply is still, it is like this, that the posterior muscles are supplied, extensor muscles are supplied by dorsal rami, and the flexor muscles are supplied by the ventral primary rami. The concept of premaxial and abaxial defines and it is concept is based on their embryological origin. So these are the references, the Langman's Medical Embryology, The Developing Human by the Keith Elmore, and Gray's Anatomy by the Susan Stranning 40th edition. Thank you very much, dear student, for a lengthy class, and best of luck for your exams, and Allah Hafiz till the next class.